Hello students. So far we have finished the microsporangium structure, anther structure, microsporogenesis, pollen structure, its development and its uses and disadvantages. So today class we are going to deal with the female reproductive part of a flower, that is about pistil or gynoecium, the structure of sporangium, as well as the development of embryo sac. Okay, so we'll start with the basic thing first. As already we know, the female reproductive part of a flower is gynoecium, or this gynoecium is also called as pistil or carpel. So this is our female reproductive part of a flower. Okay, and in the previous standards, so that is during maybe morphology of flowering plants, we have studied this pistil arrangement. Okay, so how? So based on if the flower consists of a single pistil or single carpel, such flowers what we call monocarpellary. Okay, yes, so that is a single carpel or single pistil. And if there are many carpels or more than or two or more, we can say, so such condition or such pistil condition we call it as multicarpellary. Okay, so that is many. So that multicarpellary, it may be bicarpellary, tricarpellary, tetracarpellary or pentacarpellary. Okay, two and more pistils. Okay, and the speciality of this multicarpellary. Okay, if there are, as we know, multicarpellary means the condition where there are many carpels. Sometimes these carpels are free arranged. Okay, so they are freely arranged means they all are free. Such condition we call it as apocarpus. Already we know, we have studied this. If all those carpels are fused together, okay, sometimes it may be fused also, then condition called as syncarpus. Okay, this we can see when only the flower bears multicarpels. Okay, or the gynoecium condition in such flowers are multicarpellary. In single we can't see that fusion or free arranged. Okay, wherever there is multicarpellary, this multicarpellary, where if they are 2, 3, 4 or 5, sometimes it may be free all the carpels, or sometimes it may be fused also. Okay, one example for syncarpus, because we are going to deal with the flower practically also in our practicals also. So that is the hibiscus flowers. Okay, maybe you guys have seen this condition. Okay, if there are petals are there, maybe you have seen. So this is our gynoecium. Okay, where the gynoecium ends with 4 or 5 structures. Okay, these are stigma, that is 5 in number, and all 5 stigmas are here, but we can see only one single tube, right? It means those stigma, like those pistils, are fused here. So for pentacarpellary, we can give the example hibiscus flower, where we can see multicarpellary condition, syncarpous one. Okay, that is fused. Understood? So this is what the basic thing. And always the gynoecium, or the female reproductive part of a flower, made up of 3 different sections. Okay, so that is I'll draw hibiscus gynoecium only. Okay, consists of 3 parts. So all these are, this part is called as stigma. Okay, so this long, the slender part, we call it as style, and the lower bulged portion we call it as ovary. Okay, where the megasporangium, okay, is present. That is our ovule, or it is also called as megasporangium, where the megasporogenesis takes place. The production of female gametes of flowers takes place in the ovule, or it is also called as megasporangium. We know already what is stigma, that is the landing surface for the pollen grain. Okay, style, a tubular slender like structure, okay, which is ended with the bulged portion called as ovary. Within the ovary there will be presence of placenta, okay, where these ovules are attached. Okay, so like this, consider this is the bulged ovary structure portion, okay, where there will be placenta. To this placenta, the ovules are connected like this. Okay, these are ovule, and the chamber, which is where this ovule are embedded, okay, the ovarian cavity or chamber, that we call it as locule, where this ovule are placed. And how these ovules are arranged? The ovules are always attached with the placenta. Okay, this arrangement of ovule within the ovary, okay, based on the attachment to the placenta, the term we call it as placentation. Already we have studied different kinds of placentation. It may be marginal like in peas, okay, parietal, axile, free central, and basal. Okay, these 5 types of placentation we have studied in our first year. What is placentation? The arrangement of ovules within the ovary. Okay, we call it as placentation. This is what this arrangement, and single ovule we call it as megasporangium, where that is the site of production of the female gametes of flowers. Okay, so this is what the morphological structure of female reproductive flower. We'll study in detail the ovule structure, then we'll go for megasporogenesis and development of the female gametophyte. Okay. Okay, so this is the structure of anatropous ovule. So there are different kinds of ovules. So based on the arrangement of ovule with the funicle, and always with the ratio of this is what you are going to study further, that is, this is the micropyle end, okay, we call it as. So based on this end and structures, there will be different types of ovules. But we are going to study the ovule which is present maximum, or which is shown by our maximum angiosperms, because we are studying sexual reproduction especially with the angiosperms. So in angiosperms, the ovule, okay, so this we call it as inverted ovule. This anatropous ovule is also called as inverted ovule. Okay, this is upside down. Okay, we are going to draw like this, but actually this is the above part and this is the below part, but it is arranged inverted, hence it is called as anatropous ovule. Okay, as I said, ovules are attached to the placenta, okay, by a stalk. This stalk here, it is called as, okay, so this is the stalk where they go and attach to the placenta. Okay, we call it as funicle, and this funicle is attached to the ovule, right? This ovule get attached with the funicle, the portion, this part where complete ovule get connected with the funicle or stalk. This portion we call it as hilum. And sometimes, like especially in anatropous ovule, the funicle continues with the ridge which is inserted in the ovule and will give a support to the ovule. Okay, this is nothing but a simple extension of funicle within the ovule. That extended part we call it as raphe. Okay. So next, ovule are covered by the two protective layers. Those two protective layers we call them as integuments, which in future it gives or it forms a seed coats, because the ovule itself, the fate of the ovule is seed. So hence the ovule protective covering, that is sometimes in angiosperms, there may be two integuments sometimes. Okay, so that we call it as integuments, and the future of the integuments are seed coats. And next to the integument there is a single celled, okay, not single celled, parenchymatous uninucleated cells are present. So this parenchymatous cells portion we call it as nucellus, which in turn give nutrition to the developing embryo. Okay, as in anther, like in a male reproductive part, tapetum is a layer which gives nutrition, or it acts as a nourishment thing. In the same way, in female reproductive part of a flower, that is one of the part, that is ovule, where the nucellus plays an important role to provide a nutrition for the developing embryo, and especially they are made up of parenchyma cells. Okay, they are made up of parenchyma, so hence we can call it as parenchymatous. And there will be presence of embryo sac. This is what our female gametophyte which is present, okay, in the nucellus portion. About the embryo sac, the labelings will study in detail when we are going to study the structure of embryo sac. Okay, so this is a matured ovule which contains a matured embryo sac now, which is ready to get fertilized. But we are going to study one by one this process, how this embryo sac is produced. Okay, so this is the matured stage of a ovule where there is a matured embryo sac. Understood? So this is what the structure. And one more thing, as I said, there are two ends. The above end here, it is called as micropyle end, and the opposite end, this one, we call it as chalajal end. And remember, always when there is a pollen germination, pollen tube will enter into the embryo sac to get fused with the egg is from the micropyle end itself. They get entered. Maximum percentage of plants, they undergo like fertilization, where the pollen tube get entered from the micropyle end. So this is what the structure of anatropous ovule, and it is inverted ovule. Okay, next we'll study megasporogenesis.